Thank you very much, Anita, for the kind introduction and the invitation to SSPC to be able to present some of the work we're doing in my research group at UCD. I started off at UCD in the School of Chemistry about 4 years ago, have built up my team, and by training I'm an organic chemist, but we exploit continuous flow processing routinely, and that is what I want to talk about today, but not just talking about any application in terms of flow chemistry, but specifically addressing a challenge, which is how can we discover, how can we develop new chemistry, new reactivity. Flow chemistry, as those of you who are familiar with the area know, is routinely applied in process development, and that is very important, and it's also applied in translating batch to flow processes. So these remain still important areas, but we are also interested in the discovery of new chemical reactions. So to give you a little bit of an idea what my group is interested in, we like to make drug-like compounds, bioactive compounds. They can be natural products, they can be actual drugs, or systems that have similarity with drugs. So here the first one is an example where we developed a method to make these bicyclopentane species highlighted in blue here in continuous flow. And that was the chemical challenge that needed resolving, low temperature chemistry, precipitation issues, and so on. But we're also interested in coupling downstream processes in a continuous manner. So not just performing the chemistry in flow, but quenching and extracting the crude reaction mixture. And in this application, we've also performed on top of that a solvent swap in inline chromatography. So the only job the synthetic chemist has to do at the end of the day is evaporating the solution after chromatography. And then you can characterize, you can do biological studies. So we're very interested in that area. Flow chemistry, of course, is very important in high reactive intermediates, and we are particularly interested recently in benzyne chemistry. Benzynes are, of course, not stable, so you have to make them in situ, but most methods need stoichiometric amounts of reagents. What if instead you can use light, photons as traceless reagents? So here's a method you can do that, starting from this building block, which is a bench-stable crystalline solid. You shine light at 365 nanometers at that. It fragments, giving off carbon dioxide, nitrogen, acetamide, easy to remove byproducts, and you generate benzyne, which you can engage in all sorts of different chemistries. A further area we're very interested in, again, in relation to performing continuous flow processes, coupled with photochemistry, involves photoisomerizations. For instance, you can shine light on these isoxazoles and by isomerize into oxazoles. It's a very interesting application. Or you can irradiate these amino shell cones and you can generate quinolines. These are pharmaceutically relevant heterocycles found in many drugs and also agrochemical products. But this type of transformation is also very interesting because photoisomerizations, by definition, have a 100% atom economy. There's no stoichiometric waste associated with this, no leaving groups, no protecting groups, and again, we use photons as our reagents. I won't have the time to talk about these 3 projects, but what I will do today is to talk about a project we are very excited about, shown here, which is also a photoisomerization process, but it is also a great example of generating complexity in a simple, continuous flow-based manner. So you can make these starting materials in 2 steps. Shown here on the left-hand side, you shine light on those and you generate a very complex scaffold, unprecedented scaffold, I should add, within about 7 minutes reaction time. So this is what I want to focus on in the remainder of the talk. So firstly, how did we find that? And that's something which might, or it will not be fundable as a recipe, so don't take that from me. This is research out of the blue. What we did, we had this project where we can irradiate these amino shell cones. You generate quinolines as a consequence. So that is known chemistry. You can do it in flow. It's more effective. And we published that a couple years ago. We simply asked ourselves a question: if we change this amine group to an alkyne but leave the remainder of the scaffold the same, what would happen? We didn't know the answer. We didn't even have an idea if the reaction would undergo. However, flow chemistry being straightforward and sold as a method that improves and facilitate synthesis, I would like to make the argument that we should be more open to trying ideas, even if you don't know what the outcome is. So we made the starting material, we subjected it to the same reaction conditions as for making the quinolines, meaning light of 365 nanometers, irradiation for only 7 minutes. And pleasingly, we observed that there was full conversion of substrates in a short amount of time. A new product formed, high productivity, so we can make gram quantities of these species. But the challenge in the first place was the analysis, identifying what product we had made. As listed there, we knew it was an isomer based on mass spec, but NMR showed that it was very challenging to work out what was going on. So how would you go about identifying a product? Of course, it is much handier if you have a scalable access to making this product, and because it was based on flow, that was viable, but still it was very challenging. Luckily, the product was crystalline, and we were able to get an X-ray structure of this. The X-ray structure was solved by a collaborator, Andrei Barzanov, at Durham University. And once we got that, we had finally an idea what was happening. And I want to draw your attention to the right-hand side here, where you can see a cyclobutene ring, a cyclopentene in the background, fused to this cyclopentenone. So interestingly, we managed to destroy this benzene ring here and convert it to this tricyclic scaffold. And if I draw that in 2 dimensions, this scaffold is what resulted from the benzene ring. What is also interesting is that this scaffold highlighted in blue is made by nature. It's known in this prostanoid natural product tricyclic lavulone. So in a way, although we are not making the natural product and we don't intend to make the natural product this time, but we have access to make analogs of this natural product. So having established that, of course, being organic chemists, we wanted to answer questions along the lines of how does this reaction work? It's a cascade project, so there's many individual intermediates, as you can appreciate. Somehow we go from the starting material in a few minutes to this product in high chemical yield. So what is going on? And here again, I want to make a case for advertising the use of continuous flow, not just to do chemistry, but to solve problems. The problem here is to work out what are the intermediates. So flow chemistry is great to go from A, the starting material, to Z, the final product. Why are we not able to use flow, exploit flow, to stop halfway through to make a given intermediate on demand? Normally, flow chemists are very good fine-tuning the reaction conditions to get to a final product. So how would you stop halfway through? Well, you just use suboptimal reaction conditions. So we had postulated that this scaffold here on the right-hand side, this strained ring system with the 4 or 5 fused bicycle, could arise from the cycloheptatriene. So the question now is, having postulated that, is that viable? Can we find evidence that this cycloheptatriene species is indeed involved? So again, as I outlined a minute ago, if we tweak the flow conditions using suboptimal irradiation, concentration being too high, we were able to generate this intermediate, or a intermediate, isolate that in about a yield of 50%, and characterize that. And firstly, the NMR spectrum, we got proton NMR spectrum shown here, showed in those black boxes 5 olefinic protons, which was consistent with what we proposed. So that is good, but it's not final proof. Again, how do you get final proof? Well, you try to scale your reaction. You hope that it is solid, and you get a crystal structure. Again, we were able to do that. The crystal structure is shown here. So we're able to make about 1 gram of this intermediate, and you can see the cycloheptatriene ring system quite clearly. So we can use flow chemistry for more than just making the final products. We can use it to elucidate intermediates that occur in the mechanism. The final piece of proof, which I also should mention, is, of course, not just making and characterizing the intermediate, but proving that the intermediate under the reaction conditions in question is being converted to the product. And this is the case. We used a few 100 milligrams of this compound, irradiated for a few minutes, and in quantitative yield, it formed the final structure. So Of course, there's more intermediates involved than just this cycloheptatriene, and at this point, I want to very briefly walk you through what we believe is happening. So the starting material absorbs light around 380 nanometers. We shine light from a UV LED 365, so that's a very good match. That is critical. And what we propose is that we split this alkene to this biradical. This is conventional wisdom for these kind of compounds. What is critical here is that we have the alkyne strategically placed to intercept the biradical. So the alkyne intercepts the isomerizing alkene, and that's the trick of this reaction. Once you do that, you generate a first 500 ring, you move this hydrogen, and that we propose generates a carbene, which adds across the benzene ring, giving this norcaridine ring system, and that then leads to the degradation or transformation of the benzene ring. Firstly, generating the cycloheptatriene, which were isolated, I just mentioned that, and that can undergo a second ring closure to the final product. So what is key here, again, is to notice that the conjugation is lost as we go to the final product. So highly conjugated system here in this intermediate, but we lose the conjugation as we form this less flat, more three-dimensional structure. So this is what we proposed in terms of a mechanism. Being organic chemists, of course, you need evidence for that. How do you get evidence? We are not experts in DFT calculations, but we knew experts, Cristina Trujillo at TZD and Gore Sanchez, and they collectively looked into the mechanism, and they found that what we're proposing is certainly viable, as shown here in this energy profile. So I don't want to go into too much detail, just highlighting here in the middle is the carbene. There's a pathway in red via triplet or in blue via singlet, giving us the norcaridine species, which is this compound here, and then it's just a small step to the cycloheptatriene. The cycloheptatriene, as you can see, is the lowest in energy, the most stable compound, and this explains why we can isolate and characterize this compound. In order to get to a final product, there's a huge energy barrier of about 80 kcal per mole that is insurmountable under ordinary reaction conditions. However, if you use UVA light, you can overcome this barrier and generate the final structure on the right. But I should point out that the height of the barrier and also the stability of the final product, the four-membered ring product with regards to the cycloheptatriene, is critical. And by placing substituents on the scaffold, we can affect that, as I will show in a minute. So having collaboratively proposed and provided evidence for a reaction mechanism, we moved on to exploit flow chemistry to explore the scope. And specifically, we wanted to vary all these positions as shown here in the top right-hand corner. Suffice to say that placing different substitution moieties on the left-hand aromatic ring, methylene dioxy, dimethoxy, or even plain benzene works very well. It changes the absorption characteristics, but all of them work well, as does the substitution on the alkyne partner, whether it's aliphatic or aromatic. All of these work really nicely. We also found that we can place a substituent into the para position of the right-hand aromatic ring. And here we started seeing interesting trends. For instance, if we have alkyl groups, methyl groups, for instance, also fluorine, we get the cyclobutene structure. However, if we place either electron-withdrawing nitriles or CF3 groups or electron-rich methoxy groups, we don't get the cyclobutene, we get the cycloheptatriene. Given that the electronic properties of these three substituents are very different, it's not an electronic effect, but it comes down to the height of the energy barrier. I'll show that in a minute again. But it gave us confidence that we can vary the substitution pattern on the right-hand aromatic ring as well. What we did next was a little bit more challenging. We wanted to have a second substituent here on this benzene ring, and specifically in a manner which starts from non-symmetrical scaffolds and therefore gives insight in terms of retroselectivity. Again, what we found is by placing two fluorines onto the scaffold, we get the cyclobutene, and the same was true for two methyl groups. And we got X-ray evidence for formation of some of these structures. Interestingly, if we replace one of the fluorines with a methoxy group, we get a very differentiated outcome. meaning that if the fluorine is in the para position and the methoxy in the ortho position of the form of benzene ring, we get the cyclobutene. But if we just swap them around, we get the different scaffolds. So very big differences observed for small differences in terms of the substituents. Interestingly, this is consistent with a pattern we had observed for the orthomethoxy and the paramethoxy, so there is consistency across that. The reason why, again, we switch between cyclobutene and cycloheptatriene is shown in this schematic. As I pointed out, the height of the energy barrier and the stability of the product on the right-hand side determine what scaffold you generate. So therefore, if your barrier is too high, like this one up here, which is for the nitrile, we get the 7-membered ring. If the barrier is too low, like in the case of methoxy, but the product is not very stable, you get reversibility, and therefore also the 7-membered ring product. So a last aspect I want to highlight here goes back to the retroselectivity, and I pointed out that we deliberately placed the substituents on the benzene ring in a way that it's non-symmetrical, and we only observed one regioisomer throughout this entire study. And it's always the regioisomer where the former ortho substituent is close to the carbonyl as opposed to in the other position. So what we are proposing is that in the intermediate or transition state, if you like, where we have the carbene species, the ortho substituent wants to be away from the carbene to minimize either steric or electronic repulsion. And therefore, we don't observe these species, we observe these species, these regioisomers, and that's exclusive retroselectivity we observe here. So this, as you can imagine, is a reaction which we are still exploring. It seems to give us new insights, new puzzles, new challenges with every change we make. So hopefully in the near future, we will be able to report more on this theme. But I want to come to the end of the talk and try to conclude and summarize what I showed and maybe try to give you a take-home message of where we see flow chemistry is moving, how we can exploit it in the future. Up here are some of the products, some of which I talked about in more detail, some I mentioned very briefly in the beginning, and yes, you can exploit flow chemistry to make these compounds. These compounds are fairly drug-like, so people in industry, pharmaceutical companies will recognize and acknowledge that. If a reaction is inefficient, flow chemistry will certainly help. It makes it scalable. In the context of photochemistry, which is the area we are very interested in, photochemistry does not make reactions green by default. However, we can exploit flow chemistry to add to the sustainability and greenness of these processes. So if we have good atom economy, if we can increase the concentration of these processes, we can achieve that. And again, that should have positive aspects following in terms of industrial applications. I strongly believe that flow chemistry must be used to solve problems, and it must be used to create collaborations, because that is how you get different people involved. Whether it's for DFT calculations, characterization of intermediates, X-ray, or many other things, biological studies are underway, for instance. This is a great platform. But last but not least, I would also argue that we need to start using flow chemistry as a discovery tool, not just translating batch to flow, not just for scale-up, not just for process developments. These are all important individually, but if we don't discover new things, there's soon nothing to be translated into flow. So with that, I want to leave it here, thanking the team that did all the work, specifically Mara Filippo, finally a PhD student. We're very fortunate having a lot of support from industrial sponsors, also SFI, IRC, and other partners, so big thanks to them. Thank you for your attention. Thank you for having me here, and if there's time for any questions, either now or later, I would be more than happy to answer them. Thank you.
Thank you very much, Marcus. Really beautiful example of how flow chemistry can be utilized to enhance our understanding. We've time for one quick question. Can we get a mic? Someone have a mic? You can shout, okay.
Fantastic talk. I wanted to ask, have you tried 5- and 6-membered heteroaryl species on that, and do you get the same sort of product?
Yeah, so very good question. It's very high on our list. We haven't tried them yet, so either on the left-hand side or right-hand side, but specifically on the right-hand side of the scaffold. My gut feeling is yes, there will be a reaction, but it depends, or the questions will be, is it going to be retroselective? What scaffold do we generate, if there's a thiophene or a furan or a pyrrole or a 6-membered or 7-membered heterocycle? So I have no idea. I can't even predict or suggest what will happen, but I'm sure it'll be something interesting, and we'll try that very, very soon. So, yeah.
The regioselectivity of the addition on the substituted ones, that's really tremendous, Marcus. It looks to me like something that could be exploited in terms of building them up,
Yeah, so regioselectivity is the first start, and if we start derivatizing these products, we expect diastereoselectivity, and maybe we then can look into an enantiomeric or enantioselective transformations. We want to exploit biocatalysis in this context as well. So it's great that, again, we observed by exploiting this chemistry that regioselectivity can be gained based on the intermediate being the carbene and that repulsion effect there. So, yes.
Yeah, so the confirmation probably.
Exactly, it's a confirmation, yeah.
So I think we could probably have this discussion go on for a while, but could I encourage people to perhaps talk to Marcus afterwards and continue the discussion? So thanks very much, Marcus. Tremendous presentation.